Hello. Yeah. Can you see my screen?
Yes, sir.
Yes, yes.
Okay. Uh, Last time, uh, uh, there were a couple of doubts somebody put in their chat box. Okay. Uh, Rajyoti, can you read the chat box or ask them to rewrite their doubt on the chat box and just read it to me if possible? One was, I think,
Okay,
Ankit Roy, and there was another one. There were two doubts they had put in the chat box. Just if they are there today, tell them to write their doubt again, and I'll answer them first before we go further. Okay.
okay, sir.
Ah, is there anything in the chat box by any chance?
No, sir.
Okay. Uh, Maybe I'll get started, and then as we end the lecture, when I go back to stop sharing the screen, I'll see whether I can see last time's conversation and then answer it. Is that okay?
Okay, sir.
Okay. Okay. So last time uh, we talked initially about restriction enzymes, and we said that restriction enzymes are a set of enzymes which we can use in genetic engineering because they have a very unique property of recognizing a very specific palindromic sequence and very systematically and consistently cutting that sequence. We said because of this property of restriction enzymes, if I have a gene which is present as part of the DNA, and then on either side of the gene, okay, and this is not inside the gene but outside the gene but flanking it, okay. If I have two restriction enzyme sequences, then if I use the correct restriction enzyme, for example here EcoR1, then I can cut out that piece of DNA which is containing my gene, okay. And that is the great utility of restriction enzymes, and I can do this consistently. So every time I use EcoR1, it will always cut like this and I'll always get this piece. And that is very important for me to know that I can cut very systematically where I wish to cut. Okay. After looking at that, we looked at the fact that this cut piece of DNA has to be carried into the organism where you are going to try and amplify it by what are called as vectors. Okay, and we were taking an initial example of a plasmid as a vector, and we said that plasmids are what are called as extra chromosomal pieces of circular DNA. Extra chromosomal means they are present outside of the main chromosome of the bacterial DNA, which is and these plasmids are many times seen in bacteria, and they are also seen in yeast cells and other what are called as prokaryotic organisms, and they offer the bacteria lot of advantages, and that is why a bacteria sometimes carries a plasmid or keeps the plasmid. Okay, and we said that plasmids offer a lot of advantages. For example, they might allow a bacteria to be able to conjugate and have what is called as lateral DNA transfer, or they can afford a bacteria a chance to resist the effects of an antibiotic or a poison, or they can produce compounds which can kill other bacteria, for example bacteriocins or colicins as it is called, or these plasmids might allow bacteria to produce some enzymes which allow them to digest or metabolize specific compounds which are found in the medium or in the outside of the bacteria, or they can allow a bacterium to be virulent because of their presence. And because of all of these properties which might work to the advantage of a bacteria might keep them. Okay, so this is like so what this means is, if a bacteria does not contain a plasmid, it can still survive and do all its functions. That is not the issue. But if a plasmid is present, then the bacteria gets some additional properties which might give it a survival advantage. Okay, now to use a plasmid of this type, which is actually uh, is able to come into a bacterium, replicate just like the bacterial DNA can replicate, is an advantage in what is called a genetic engineering, and it is used very specifically because of this unique property the plasmid has, that it is able to get into a specific bacterium, multiply in that bacterium. When the bacteria divides, it also divides and it gets passed on to other bacteria. Now, for a plasmid to work as what we call as a cloning vector, that means I want this vector to do my job for me, carry a piece of DNA that I'm interested in into a specific bacterium, and then not only that, stay in that bacterium, multiply along with that bacterium and make multiple copies so that I'm able to clone or amplify the DNA. So the property that you want from it are it should be self-replicating inside the cell, and we already said most of the plasmids have that property. It must possess unique restriction site for restriction enzymes. Okay, the reason for this requirement, which will be evident in a slide or two, is that because I need to add my DNA to this vector, I should be able to cut this vector in a very systematic fashion so that I can put a piece of DNA inside it. Okay, and therefore for that I need what are called as very specific restriction sites. Also, when I take this plasmid and I add a piece of DNA, it should not interfere with the function of the vector, okay, in terms of being able to replicate. It can affect some other property, but at least it should not anytime affect the ability to replicate. In other words, once I put the DNA fragment into the vector and I put it back into the bacteria, it will be able to multiply. Okay. Also, if possible, and I didn't say even possible, almost necessarily, it should have some marker which I can later use for identification of a cell which actually carries the plasmid. Okay, in other words, remember different plasmids have different properties. So I want a property in a plasmid such that it will allow me later to figure out whether a bacteria contains the plasmid or not. For example, an antibiotic resistance gene which is normally absent in the original bacteria. Okay, so let us say I have a plasmid which allows resistance to antibiotic like penicillin. A normal bacteria, when exposed to penicillin, might die, but if I put this plasmid in it, then that bacteria will survive, and that will tell me that the bacteria is carrying the plasmid because it is now not able to be killed by penicillin. And finally, it should be able to be put as well as isolated from the host cell easily. Okay, now there are lot of plasmids that occur in nature, okay, naturally, okay. But the problem with this naturally occurring plasmids is they are there to carry out the function which is not related to our intent, that is wanting to do genetic engineering. So many times these plasmids are of no use to us. Although they have some properties, they don't have all the properties that you wish. So what people have done over the years is actually identified some very special plasmids which have certain properties which make them much easier to use in genetic engineering. Okay, and in that regard, one very famous plasmid uh, is referred to by the name PBr322. Okay, now here in this name, this P refers to the fact that it is a plasmid. Then Br are two initials of the scientists who actually came up with this plasmid, who actually manufactured or synthesize this plasmid. B stands for Bolivar, Bolivar, and R stands for Rodrigues, okay, Rodrigues, sometimes also pronounced as Rodriguez. Okay, so PBR stands for Plasmid Bolivar Rodriguez, and 322 is a number which has significance for them. Maybe it's a page number of their research book, or it is the 322nd plasmid that they designed, or experiment that they did, whatever it is. Okay, but it is very famously called as PBR 322. Now this plasmid is a circular double-stranded plasmid. Okay, I've shown only one strand here, but actually it is double-stranded, and it contains a total of 4,361 base pairs. Okay, so let us say I were to start from this point, and if I were to keep going clockwise all the way and come back to the same point, I would have passed what is called as 4,361 base pairs. Okay. Now this plasmid has some unique properties which make it very suitable for doing what is called as genetic engineering. Okay, and it works very well as a vector. Now, one property that all plasmids have, and also PBR 322 has, is that it has what is called the ORI. ORI is short for origin of replication. Okay, so whenever this plasmid is present in a bacterium, okay, and it has to be replicated, the process of replication always starts at this point, and then it goes in either direction. Remember, bidirectional replication, it goes in either direction and it allows the plasmid to get replicated. Okay, so the first thing that is necessary and the plasmid has is the origin of replication. The other two interesting properties it has is that it has got two genes. Okay, one gene here is called as AMP, and the other gene here is called as TET. Okay, now what do these two genes do? This gene here produces a protein which allows a bacteria which has this plasmid to be resistant to this antibiotic ampicillin. Okay, so in some books they'll write this as AMP and superscript R, and they will call this an ampicillin resistance gene. Okay, which means when this gene is transcribed, you make protein out of it. That protein allows me to show resistance to this ampicillin. In other words, now the bacteria which contains this plasmid will be able to grow if the media contains ampicillin. Not only that, it also provides the bacteria an added resistance. This gene here codes for another protein which allows the bacterium which carries this plasmid to be even resistant to tetracycline. Okay, remember, ampicillin and tetracycline are two antibiotics which belong to different classes. This is what is called as beta-lactam antibiotics, and these are antibiotics which are of the anthracycline type or anthracene type. Okay, so if this gene is present and a protein is produced when this gene is being expressed, that protein allows the bacterium which carries this plasmid to be also resistant to tetracycline. What does this mean? If I have a bacteria which contains this PBR 322 plasmid, it will be able to grow even if you put ampicillin in the media or tetracycline in the media or both in the media, because it provides that advantage to that bacteria. Okay. Now, also shown is something very interesting. If you look here, there are names of lot of restriction enzymes. This is one restriction enzyme called PST1. This is what we saw yesterday, EcoR1. Then there is BAMH1, SAL, EcoR5, HIN3, and so on and so forth. What does this mean? In this plasmid, at various places, there are very specific palindromic sequences which are recognized by these restriction enzymes. which means that if I were to take a plasmid PBr 322 and treat it with eco R1, it will cut the double strand here. Okay, and remember, it will cut on both the strands, and what will simply happen is that this circular plasmid will now become linear because it has been cut. So I'll get one linear piece instead of a circular piece. In the same way, if I were to use BAM-H1, it will cut exactly here, and then it will open up the plasmid, but slightly differently, but again you will get a linear plasmid. And in the same manner, if I were to take this plasmid and treat it with both EcoR1 and BAM-H1, then I'll end up actually getting two pieces. One piece will be this one, which is cut here, and the other piece will be the remaining, which is long like this. Okay? Is that clear? Now, what this means is that I can. Hello? Yeah. Is there a question? I can't hear.
No, sir.
I can't hear. I can't hear. Hello?
No, sir, it
I
was a
can't hear.
disturbance.
Okay, okay. Okay. So what this means is I can use this plasmid and very systematically, using whichever restriction enzyme I wish, cut it exactly where I want. Okay. For example, if I use PST1, I would cut right here, right in the middle of this gene for ampicillin. Or if I use BAM-H1, I would cut right here in the middle of the gene for tetracycline, and so on and so forth. Okay. Just like PBr 322, there is another plasmid which is called as PUC. Okay, or sometimes people read it as PUC18. Okay, here P stands for again plasmid, and UC stands for University of California, where this plasmid was what you call as invented or designed. Look at this plasmid. It is 2.7 kilobases long, which means it has got 2,700 base pair. So it is slightly smaller than PBr, kind of a little more than half the size. Okay, but it has got some properties which are very similar. It has got an origin of replication. It also has got a gene or a sequence which allows it to be resistant to ampicillin. But then it has another gene which is responsible for producing an enzyme called as galactosidase. Okay, this word LAC-Z stands for the name of the gene which produces an enzyme called as galactosidase. And what does galactosidase do normally? It can take this disaccharide lactose and break it down to galactose and glucose, okay, which allows the bacteria to use lactose as a source of energy. Okay, and in this LAC-Z gene are lot of palindromic sequences which are very specific for this many different types of restriction enzymes. Okay, and they are present right here in the middle. So I can use any one of them to cut the gene somewhere in this region. Okay, everyone. So now, for a little while, I'm going to talk a little bit theoretical before we go back to the practical side. Okay, so a little bit of what is called as general notes. So plasmids are extra chromosomal DNA. They tend to be in the range of 5 kilobases to very small, like only or fairly big, I should say, 400 kilobases. Okay, which means 5,000 to 400,000. Okay, they get inserted into bacterial cells by a process called transformation. We'll see what this is. Now, this is very important. Plasmid, when they are used as vectors, can be used to put pieces of DNA which are up to what is called as 10 kilo base pairs. Okay, when I mean up to 10 kilo base pairs, what I mean is at the most I can put 10,000 kilobases. But you have to remember that how much you can put depends actually on the original size. Okay, so for example, if I have a plasmid which is only 5 kilobases and I try to put 10 kilobases in it, I'll actually get plasmid less and foreign DNA more. Okay, so these type of plasmids will take up much less DNA than 10 kilobases, but something like this plasmid, which is 400 kilobases, can probably take up 10. So read this correctly. It doesn't mean it can take up this much. It can take up to this much. Okay, and most plasmids carry a marker gene. Okay, as I told you, like for ampicillin resistance, etc., which allows the bacteria that contains the plasmid to grow in the presence of that antibiotic. Just like we can use plasmids as vectors to carry DNA, people have also used viruses to carry DNA. For example, many bacteriophages are used to carry foreign DNA. And how do they carry DNA? They carry the DNA by infecting the bacteria like they normally do. But what we do typically is, if I want to take a foreign DNA in, I modify the genetic sequence of the virus itself and put the piece of DNA in the viral genome in such a way that the virus still is functional, so that later when it infects a bacteria, it will deliver my DNA also. But the advantage of using bacteriophage as a vector is that it can take up sizes which are slightly bigger, okay, which is up to 20 kilo bases. Okay. Similarly, if I want to insert even bigger pieces of DNA, okay, then the vector I use is called as a bacterial artificial chromosome. Okay, these are plasmids designed to take up 75 to 300 kilobases. Okay, again, they are no different from any other plasmid, that they have antibiotic resistance, they have origin of replication. But what is important is they are able to carry larger pieces of DNA, and they are made artificially in the lab. They don't occur naturally. That's why they are called as bacterial artificial chromosomes. Actually, we should call them bacterial artificial plasmids also. Okay. Similarly, we have what is called as yeast artificial chromosomes. Okay. Now, these are very specific for carrying DNA into yeast. And what is the advantage? They can carry even bigger pieces. See, 100 kilobases to 3000. Remember, plasmids carried up to 10. Then we had your bacteriophages, which was carrying up to 20. This was carrying up to 300. And now I have up to 3000. Okay, and these are using for cloning very large pieces of DNA, and sometimes they're using for use for other purposes, which is called as genomic sequencing of genomes, etc. But they have the advantage that they also can carry what are called as DNA sequences into eukaryotic systems rather than prokaryotic systems, which has certain advantages in genetic engineering. And not to let finish, you can also have human artificial chromosomes. Okay, so this is very specific for taking DNA into human cells or mammalian cells in general. And look at how much DNA they can carry, 6 to 10 mega. Okay, now remember, previously it was 3 kilobases, which means it is 3 megabases. Okay, 3000 kilobases means 3 mega. This can take 6 to 10. Okay, now these are able to take large pieces of DNA. Okay, now all of these can potentially be used as vectors. Okay, so I've introduced to 5 types of vectors: plasmids, bacteriophages, BACs as they call it, bacterial artificial chromosomes, YACs or yeast artificial chromosomes, or HACs, human artificial chromosomes. Now remember, this is not everything. There are even other types of vectors, like what are called as cosmids, phagemids, etc., etc. But since we are just talking about introductory genetic engineering, this is enough. What is important to realize is I can use different vectors such that they are specific for different types of cells, and also for different pieces of DNA in terms of size. Okay. Now, we have talked about restriction enzymes, and we have talked about what are called as vectors. Okay. Now, I'm going to do something very interesting. Let us say I have a piece of DNA, and I cut it with a restriction enzyme. Okay, in this case, let us say a restriction enzyme which recognizes the sequence GAA-TTC, which is also palindromic.
Hello, sir.
Yeah.
Sir, sorry to disturb you, but
Yeah.
can you please change the slide to the yak yeast artificial chromosome?
Yeah. There we go.
I had a doubt there.
Yeah, there we go. Yeah.
Sir, it's written here that
Hmm.
yak produces chimeric effects.
Hmm.
What exactly that means?
Okay. What sometimes it will do is yeast artificial chromosomes tend to do recombination. Okay, so sometimes they will interact with the yeast ka own chromosomes, and they will get merged, and they can kind of take pieces from it and give pieces from it, and sometimes that results in effects which you don't predict. So chimera is like what we typically call in mythology as head of a sheep and body of a goat, or head of a tiger, body of a horse. Okay, so if I start with a yeast artificial chromosome, and then it is able to take pieces from somewhere else and put it inside it, or give pieces of it to somebody else, it will give what is called as chimeric effects. And when that happens, you don't know exactly what is happening, and which is not good. And that's why this terminology that they are less stable compared to bacterial artificial chromosomes. Okay, which is a little more stable. They don't do this recombination events. Okay, is that clear?
Okay, sir. Thank you.
Okay.
Yes, sir.
So coming back to this slide, as I said, that if I have a piece of DNA and I treat it with a restriction enzyme which cuts it, and I get these sticky ends. Okay. Now imagine that I have another piece of DNA. Okay, a piece of DNA which is actually structure wise different than this piece. The only similarity between that piece and this piece is that that piece also has a recognition site for the same restriction enzyme. Okay. Now let us say, for argument, that I were to take that DNA and cut it with the same restriction enzyme. I am going to generate this piece. Okay, because this piece is one of the pieces which is generated by the restriction enzyme because of recognition of this palindromic sequence, which now is also present in this DNA. Now, if I were to take this test tube ka mixture which contains these two things, and this test tube ka mixture which will also contain a piece which is like this and a piece which is like this, but which is different elsewhere, what might happen is that because of this similarity of this end and this end, I might find that if I put them together, this piece which is from this DNA might actually start interacting with this piece which is from another DNA, which means I can actually recombine this with this and replace the original DNA. Okay? And if I now say, for argument, treat this with DNA ligase, which will again reseal these ends, then what will happen? I'll get now a new piece of DNA where this end is from this DNA, while this end is from other DNA, and this was possible because of this similarity in the sticky ends. Okay? Now, and this is the
So,
philosophy. Yeah.
what about if the nucleotide, the bases apart from the palindromic sequences are not same?
It doesn't matter. Okay, all I'm interested is in this part where they all combine together. Okay, now it will be more clear in the next slide. Okay, so I just wanted to put this concept here. Now I'm going to go in terms of what exactly happens. Okay, remember that slide I showed you where I said that you have a target gene and you have a vector, and you are going to recombine them and make a recombinant plasmid. Okay, so now imagine for a second that I have a long piece of DNA where this is the target gene, which is shown here with the arrow. Okay, and flanking them, shown in violet, are two sequences which are palindromic. Okay, and these sequences are shown here in a amplified manner, or what is called as magnified manner, is GAA-TTC and GAA-TTC. Okay, which means that if I were to take this target gene and treat it with EcoR1, I'll get one cut here, I'll get another cut here, which is between GA, I'll cut one more cut here, and I'll get another cut here. So whatever be the length of this DNA, I will generate this small piece, which is my target gene, flanked by two sticky ends, which are generated because of E. coli cutting. Okay. Now, let us presume that I have a plasmid which also has one site which is palindromic, but which has the same sequence as this one. If I were to take this plasmid now and I cut that with EcoR1, what will happen? It will cut it here and it will cut it here. And when that happens, this part and this part will come apart. So from a circular plasmid, I'll get a straight plasmid, but which ends are like this. Okay, they will have overhangs which are the same, TTAA, TTAA, and that will be at the 5' end. Okay, now what I just said is shown here. Okay, so I take this, cut it with EcoR1, I will generate this piece, I will generate this piece, and I will generate this middle piece. And let us say I take a plasmid and I cut it with restriction enzyme, same restriction enzyme, which recognizes the same sequence. This plasmid will open up, and the opened up plasmid will look somewhat like this. Okay. Now, if if you have the insight, you will realize that, see, this piece here, which is TTAA, is actually complementary to this TTAA, while this end, which is TTAA, is complementary to this, which is AATT, which means that this piece, if it is able to slip here, what will happen is after this piece slips in, this end will actually be complementary to this, and this end will be complementary to this. Okay. What that means is that if I were to mix all the contents of this test tube, where I have taken this target gene and cut it up, and the contents of the plasmid containing tube, which I have broken up with EcoR1, I mix them, there's a chance that what I can get is a plasmid which now contains this target gene, which is now sitting pretty here. Okay. Now all I have to do is treat this system with ligase. Now if I do that, and I know what does ligase do, every time there is a break and there is a OH and a phosphate, it uses ATP and makes a phosphodiester bond. So what will now happen if I treat this with ligase? I'll get a piece of DNA which is now perfectly integrated. Okay, and I've got a recombinant plasmid which is now carrying my gene of interest. Okay, are you seeing how I have made the recombinant plasmid? The only condition for this to work is that my target gene should have two palindromic sequences flanking it, recognized by a given restriction enzyme, and my plasmid should also be cut into one piece by the same restriction enzyme, and then if I mix the cut pieces, then there's every chance of me getting this. Okay. Now, although what I said is very, very easy, what you should also realize is something like this. Okay. Now let us say I do this. Okay. Now remember, I am not doing this with one molecule. I am doing this with a molecule which is present in million copies here, so that when I cut, I get a bunch of pieces which are all like this. Okay. It's like, for example, sometimes you write a synthetic reaction and we say acid reacting with an alcohol gives an ester, and we write RCOOH plus ROH giving RCOOR, and we say this is a reaction of acid reacting with an alcohol to give an ester. And even when we want to make an ester, we say you can take this acid and you can take this alcohol and you'll get the ester. But what we actually mean is that you can take a whole population of acids and you can take a whole population of alcohols, and if you combine them together, you'll get a whole population of esters. Okay, which means that when I'm talking about this, you have to realize that I'm not talking about a single copy of this and a single copy of this, giving me this and this, so that later when I mix it, I'll get only one of this. In reality, what happens is I'll have multiple copies of this, and I'll have multiple copies of plasmid which I have cut. And I'm looking for this event to occur at random, where this piece will end up here. Now you have to remember, if there are multiple copies of this, each copy also has ends which are very complementary, right? So imagine there are two such pieces, okay? Two pieces of this target gene. The second piece, which looks exactly like this, can actually come and sit here, because its end AATT is exactly complementary to this. Likewise, if I have a whole number of plasmids, remember that other plasmid also has got an end which is like this. So in reality, what happens is when I try to do this experiment, a lot of things happen. Sometimes I'll get vectors joining to each other and I get a whole long piece of just vector, okay? Or sometimes I'll get just that cut DNA combining with each other, and when I treat with ligase, I get one long piece which is joined, but it is only my foreign target gene. Okay, it is not recombination. And the third, and what I want, is the event which is a recombination event, where this has correctly inserted into this. Okay, when all of these things can potentially happen, how will I know whether I have generated this piece or only this piece or only this piece? Okay, now, and that is where that
Hello.
plasma. Yeah. Hello.
Hello,
Yeah.
sir. I had a doubt
Mm-hmm.
that
Yeah.
if a plasmid is cut at multiple points in the
Mm.
in its sequence,
Yeah.
then there would be a very low chance of getting the original plasmid back.
Correct. But see, remember, if you look at all the diagrams I've shown you, no, I've been very intelligent. Okay. I do not cut the plasmid at multiple sites. I cut it only at one place. Okay. So at the most, I will get one plasmid cut and I'll get one piece. And if you look at the vectors also, no, which I showed you, look at how the vectors are. E. KRI side, there is only one. HIND1, there is only one. Eco R5, there is only one. And that is the advantage of these plasmids. So if I treat with Eco R1, no, it will cut only here. It will not cut anywhere else. Likewise, if you look at PUC, it has got many restriction sites, but there is no repetition. Eco R1 only cuts at one place. It doesn't cut anywhere else. Okay, and that is what something you have to realize, that when I use the vector, I use a vector which is such that it can be cut by the restriction enzyme only at one point. If I make the mistake of choosing a plasmid which has got multiple sites, then I'll get a whole bunch of pieces which may not ever come together. It will be like Humpty Dumpty. समझा ना? Humpty Dumpty का story याद है ना? It was an egg which fell and nobody could put it back together. Okay, it will be a situation like that. But if I cut it only once, then the only thing that can happen is that plasmid can just come back. Okay, either as is or with this piece in between, or two or more plasmids can join together and all those events can occur. But I will always be able to reconstruct the plasmid. It will not be in shatters. It will not be in bits and pieces. Is that clear? Did I answer your question?
Yes.
So that is why I make this statement. So if I cut a plasmid and it joins to itself, I'll just get a bigger plasmid. Or if I cut a target gene and it simply joins to itself, I'll get a bigger target gene with lot of these targets. But what I'm interested is in this going into this, which means I want this combination event, okay, which we call as a recombinant plasmid, which looks somewhat like this. This is the original plasmid, this is a new piece which has come, and I've got a recombinant plasmid. Now, and how do I identify what has happened? And typically what we do in real life is we never try to separate these things. we let them happen, okay, because they are almost impossible to separate, okay. So typically what is done is I take a whole bunch of vector molecules, I take a whole bunch of my DNA which I have cut out, I simply mix them together, I will treat them with ligase, and I'll allow all these events to occur at random, okay, I don't care in a sense. But what I will do is I will select for which bacteria has taken up this type of plasmid by the unique property the plasmids have, okay. Now I'll show you what happens a little later, but before that, what is important is after I have made these pieces and I treated them with ligase, how do I put it back into a bacterium, okay, and that is done by a process called as transformation, okay. Now what is transformation? Transformation is a process by which you introduce foreign DNA into a host cell, okay. In this example, recombinant plasmid into a bacterium. There are several methods for doing it, okay. Some bacteria have a natural ability to actually take up plasmids which are present in the medium, okay. Now if you have a bacterium of that type which you are going to use for amplification, God be with you, you are lucky, okay. Usually many bacteria don't have that property, only a few of them have that. So what we tend to do is do what is called as heat shock. What we do is heat the culture of bacteria to about 45 degrees or 50 degrees. At that temperature, you actually create what are called as little pores in the bacterium because of this heat shock. And then as soon as you take it, you don't keep it at a high temperature for a long time. You take it to a higher temperature, allow these pores to occur, and then add your plasmid and then cool it immediately. Okay, what will happen many times is many bacteria will die because of this increase in temperature, while some might take up DNA through the little pores that have occurred and then even survive the heat shock, and then they will carry the plasmid. Sometimes people use calcium phosphate precipitation. What this is remember calcium is a divalent cation. It has got a lot of affinity for DNA, which has got phosphate as a backbone. And DNA tends to form a little precipitate on calcium phosphate, or what we call as chalk, a chalk variant as it is called. Okay, and then now in the media you have these little pieces of calcium phosphate on which DNA is precipitated, and many times bacteria will take them up by endocytosis. And when they are taken up by endocytosis, the DNA gets into the bacterium, and then you have the plasmid going into the bacterium. So just like you can do heat shock, you can also do what is called as electroporation or sonoporation. Okay, as the name suggests, here you use electricity to make little holes in the bacteria by shocking them, or you use sound waves, many times ultrasound waves, to create holes in the bacteria, and you do it for a very short time. Okay, and soon as you have done that, what you do is you add your plasmid DNA or recombinant DNA, and as the bacteria tries to survive these holes, some of these plasmids will go through the holes into the bacterium. The bacteria might seal itself up and survive the electroporation, and after it does so, it might have the plasmid inside itself. Or if you have cells which are fairly big, like human cells or animal cells, or many times very hardened cells like plant cells, you need to introduce DNA in more interesting fashions. For example, here you can actually inject the DNA into the cell because the cell is big enough. Or in case of plants, you fire very small bullets which are coated with DNA into the plant or the tissue, and as the bullet pierces, it will go through the cell wall of the plant and go into the cell and deliver the DNA, and hopefully as that is happening, the cell survives. Okay, so there are various methods by which you can what is called as transform a bacteria or introduce DNA into a bacteria. Okay, the most commonly used methods are generally heat shock and electroporation in bacteria, and you use microinjection and ballistics generally for animal cells and in this case plant cells. Okay, now Imagine for a moment now that you have made this recombinant which contains amongst other things the correct combination of foreign DNA added to the vector, and you have taken this entire mixture and you have tried to introduce into this bacteria at random. Okay, so now different bacteria might take up different things. Okay, and what you are interested in is the bacterium which has taken up the molecule of interest. That means all of these have gone in at randomly in different bacteria, and you are interested in the bacteria which carries this. Okay, so now remember I told you this is where your vector ka properties become very important. Okay, so let us say, for example, I take PBR 322 and I cut it with PST1. Okay, if I cut it with PST1, what will happen? I'll make a cut here. Okay, now imagine that the foreign DNA that I'm interested in putting into this vector is also cut out with PST1. Then what will happen is that piece of DNA will have two ends which are such that they are complementary to the ends that have been generated here. So when I mix that DNA with PBR 322, that piece of foreign DNA will end up and come and sit here. Okay, and when it sits here, what it will do here is it will what is called in a sense tamper with this original gene for ampicillin resistance. So it will actually damage the gene, and the way it has damaged the gene is by putting itself in the middle of this original gene. So now in the recombinant plasmid, what you'll find is this black line, then the foreign DNA, and this line will then continue, which means this gene is now disrupted or damaged. Now see what happens because of that. Okay, now because I have disrupted this here, now whichever bacteria carries this type of plasmid will only be resistant to tetracycline. It will not be able to show resistance to ampicillin. The reason simply is because the gene for ampicillin resistance, which produces a very specific protein which allows the bacteria to be resistant to ampicillin, now doesn't produce that protein. It produces some defective protein or doesn't produce anything, which means a plasmid which is of a recombinant type will be sensitive to ampicillin and resistant to tetracycline. Okay. Now remember the original plasmid, right? The original plasmid. Which is the original plasmid? A plasmid in which nothing has been inserted. Okay. That plasmid is resistant to ampicillin. Is it or is it not? It is. Okay. Now, and the recombinant plasmid is the one which is, as they say, sensitive to tetracycline. Okay. Now I can do the same thing with respect to this gene also. So wherever I do the insertion, I will lose that. Okay. Now look at this example. I've got these two here, where what I've done is I've damaged this gene. Okay, and this gene turns out to be the gene for tetracycline. Okay, which means if I were to look at this fellow here, he will be resistant to ampicillin, sensitive to tetracycline. While this, which has got no change relative to the original one, will be resistant to both ampicillin and tetracycline. Okay. Now, think of this. I have taken a vector which shows resistant to both ampicillin and tetracycline, and for argument, I have introduced the gene which I am interested in into one of these sequences in such a way that I destroy the ability of the plasmid to be resistant to one of the antibiotics. Okay, what I have done is something like this. Okay, so if you look at this slide, no, which is very interesting, what I have done in this case is I am showing you an example where essentially what I have done is introduce the gene here. By introducing the gene here, what has happened? the recombinant plasmid is no longer able to show what is called as sensitivity to tetracycline, okay, or resistant to tetracycline. It is now sensitive. Okay, now imagine that has happened. Okay, now after I introduce the mixture, okay, and remember this mixture here into bacteria, what I will find is all the bacteria which take up just the original plasmid, okay, or combinations of the original plasmid will show a very interesting property. They will be resistant to both ampicillin as well as tetracycline. Okay. While this fellow here, because I have inserted the foreign DNA very specifically, will be resistant to ampicillin, but it will be sensitive to tetracycline. Okay. I use that to my advantage. Okay. So what I do is I transform the bacteria and I allow them all to grow in the medium which contains ampicillin. Okay. Now I know that in this situation, all the colonies that I will get are colonies of cells which are carrying plasmids which are able to resist ampicillin. And this includes those bacteria which contain the original vector and also the bacteria which contain the recombinant vector. Then I do something very interesting. I take this colony and then using what is called as a a sterile pad or a sterile filter paper, I dab it on this colony and I take that dabbing and put it on a fresh petri plate which contains medium which has tetracycline in it. Okay. So what will happen now is I will get a few bacteria from each of these colonies which are now transferred very systematically to this plate, but the difference is that this plate contains tetracycline. So what will happen now? As all of these colonies which are here start to grow at the same location on this plate, only those fellows which are able to resist tetracycline will be able to grow. Okay. All the others which are sensitive to tetracycline will perish. Okay. For example, let us say I did this experiment and I find that now in this colony, in this petri plate, only 3 of these are growing. What is this is telling me is these 3 bacteria are resistant to both ampicillin as well as tetracycline. So what I do is I go back to my master plate from which I had only taken a little dabbing and I put it here, and I look at the remaining 3, the ones which disappeared here. I will realize that all 3 of them are the ones which contain recombinant plasmid because they are showing resistant to ampicillin, but the minute you add tetracycline, they are dying. Okay. Now this way of what is called as detecting a recombinant plasmid as it is present in the bacteria is called detection by looking for insertional inactivation. Okay. What I mean when I said insertional inactivation is when I inserted a foreign DNA into this gene, I inactivated that gene. And this protocol that I just talked about is called as replica plating. See what you have done, you have grown all the cultures in ampicillin first, and then you have made a replica plate by taking a pad or a filter paper, dabbing it, and in the same orientation putting it on a fresh petri plate. So this petri plate is actually a replica of this in terms of where the colonies are. The only difference is it contains tetracycline. Okay. So this is how you end up selecting all those bacteria which end up containing that DNA or that plasmid which carries your DNA. Now all you have to do is take those plasmids and grow them in culture, and you'll get large amounts of these bacteria as they multiply, and as they multiply the plasmid will also multiply, and this culture is always grown in ampicillin. Remember, these fellows are resistant to ampicillin, so you grow in ampicillin so that no other thing grows. Okay, and this recombinant plasmid goes selectively. I should not make the mistake of putting tetracycline here. What will happen if I do that? I'll just kill all the bacteria which contain recombinant plasmid because they are now sensitive to tetracycline. Okay, so I shouldn't make that mistake. Huh? So I put antibiotic ampicillin here. They will grow very nicely, and then later on I can take out the plasmid and cut out the DNA, and I'll get my clones. Okay, and now what do you call as cloned my DNA. Okay? So I'm going to stop here for the day. Any doubts? You ask me now. Okay, and then at this point I'll stop my screen sharing. Hello, can anyone hear me?
Yes, sir.
Yeah.
Yes, sir.
Uh, so yeah, tell me.
Sir, there are doubts in chat box.
Okay. Let me look at the chat box now. Okay. So, let me start from the beginning. What if you want to cut a palindromic gene out? Okay. Now, genes are rarely palindromic. Okay. A gene is a large sequence of DNA which codes for a protein. Okay. For example, you can imagine if I have a protein which has got about 100 or 200 amino acids, the minimum length of gene that I need, realizing that three, basis code for one amino acid is a DNA which is a length of 300 or 400. It is very difficult to have such a long sequence which is palindromic. Okay, so we rarely have genes which are palindromic. What we end up having is genes which are surrounded on either side by small sections which are palindromic, and that is what we cut out. Okay, so that was a doubt by Mohare. Okay, here only colony characteristics can be used as markers. No, not necessarily, but that is one of them. The other way is to actually, grow the bacterium and different bacteria and actually check for the sequence of DNA itself, whether a certain piece of DNA is there in the bacteria which is growing. Okay, that is a little difficult to do. Okay, that's why people use colony characteristic, which is much easier. Okay. Now, to seal the ends after inserting the required genes that are produced by restriction enzyme, uh, ligases can be used. Yeah, we use ligase. Okay. Replica plating is used just to identify the recombinant bacteria for plasmid extraction. Okay. Yeah, correct. Uh, Basically, if I do replica plating in the second step, I will know which are the colonies containing the recombinant bacterium. And what I will do after that is not directly extract the plasmid from them. First grow them. Okay, make millions of copies of them, and then from that I can extract the plasmid. Okay. Replica plating is also used to identify sensitivity. Yeah, it is. Because when you do replica plating, in the medium you put an antibiotic, right? So you'll know whether a bacterium is sensitive to that or resistant to that. Okay. So I hope I have answered all the questions. Any other question that you wish? Okay. Okay. So what I'm going to do is stop sharing, and I'll here I stop recording, and then